evolved STEM. All of you have seen that there was an assignment that was on Google Classroom. And today we're going to start walking through it. So she's going to take over, but first we're going to do a quick poll to see if you were able to access all the material, walk through it, how much time you spent on it. So I'm going to launch a quick poll. Please be honest. I think this is the first time anyone's tried to do this remotely, and Dr. Matela is trying to learn a lot about the process to make it better as going forward too. And likewise, you know, as you have little small breakout groups today, or we come all back together, if you have thoughts, suggestions on this material, or you needed more explanation, or whatever you wanted, please feel free to share that with us. I know Dr. Matela is working very hard to try to make this material accessible to everyone. So that'd be really, really beneficial. So I'm going to launch a poll. If you guys could just take a couple of seconds to answer the questions honestly, please do. So we're getting a nice mix. Ivy, can you see the results as they're coming in?
I can.
Great. So it looks like most people have done something, but most of it was some and not all. And that's good to know. And you're getting a little bit of an understanding. Let me see how many. 1, 2, 3, 4, 5, 6, 7, 8, 9. I think that's everyone. So why don't we end this poll and you guys can see where you fit in. So thanks for all of you for starting to interact with the materials. And this is good to know. So you're through some of the materials and they're spending under an hour on it. So there you go, Abby. That gives you a little bit of information about how this is going forward. Oh, I didn't share the results, so you guys can see it. I will, though. And with that, I'm going to turn it over to Dr. Matela, and she's going to start walking us through this cool experiment that you aren't going to get to do in person, but you're going to get to learn about it virtually. So, Abby?
All right. Okay, can everyone see the screen?
Yes.
All right, so I thought what we would do today is, since you guys, most of you have been able to go through the materials, but maybe not look at everything or understand everything in detail, that I would just kind of give an overview of this introductory material, and then we would just take some breaks throughout to kind of work in groups to answer some questions for discussion, and then come back and talk about them. And I think there was actually, we'll see if we get through everything today, because I feel like there was a lot of material for this first section, so we might move part of this, you know, we'll kind of just end at the end of the hour and then move some of it into tomorrow. Okay, so the beginning is just looking at an overview of the experiment. And I love to ask questions as I go along, but I will, so I feel like most people have heard of Darwin's finches, right? And I can see some of you, but how many people, if you can just raise your hand, have heard of the finches and their adaptations with changing beak sizes to be able to eat different food sources. So I think that's a really common example that we share in classrooms of how evolution can result in these in adaptations to different environmental conditions. But of course, you know, finch evolution, we can't watch that. You know, it's not observable in a reasonable amount of time. And so in our lab, what we've looked at is bacterial adaptations, and in particular, When we select for bacteria to adapt to form biofilms, what we find is that we get a lot of diversity. So these are bacterial colonies. And this bacteria is a different one that's used in the laboratory experiment. It's called Burkholderia senescapacea. It's one we use in our lab, but not in the classroom. And the ancestor has this little smooth round colony phenotype. But when we select for that ancestor to produce biofilms, then we can get a variety of different colony morphologies arise over time.
I have a question about that last picture. So the ancestor and the studded ones, like they have really similar shapes. Like they look like different colors, but that just might be the type of imaging that was done, because I know that's really small. So how did you guys tell the difference since they're the same shape?
So I think that's a great question, because, you know, to my eye too, I feel like there's really similar looking. And I think the difference, which is probably easier to see on a plate, I don't work with Burkholderia, but other members of the lab do, is you can see that it's a little like kind of wrinkly, you know, that top shape is somewhat different. And maybe the other thing that's good that you're bringing up to point out is that not only when we plate the bacteria do the colony morphologies look somewhat different, that not as much as these other, the ruffled spreader and the wrinklies, but because they're grown in test tubes, what we're looking at here is how they form biofilm on the side of the test tubes as they grow. So you can see that that's actually different, right, across these different morphologies. So if you grew them alone, you know, studded makes this kind of short little round layer of biofilm, where the ruffled spreader is more spread out, and the wrinkly also more spread out and higher production. Does that answer your question? Yeah,
Yeah,
you know,
it does. Thank you.
yeah, and I will add in, I know we're gonna have a little time for discussion questions in groups, but please feel free to interrupt and ask questions as we go along through this. So this, how many, I should have asked that in particular. I know you guys, most of you were able to watch some of the videos. Did everybody watch this particular video? Like, what are biofilms? Yeah, for most people. Yeah, this is probably the most fun video, and we won't go through it just for time's sake, but if you hadn't watched it before, I would suggest looking at it because I think it's a good one. And really the point, if you had not heard of biofilms or known about them, is that what biofilms are communities of bacteria. So bacteria, most bacteria actually live in these close-knit communities where they'll produce an extracellular kind of slimy matrix that protects them from predation and outside attack from different things such as antibiotics. And how that happens is that individual bacteria will attach to a surface, and then as those bacteria reproduce, they'll start to produce this slimy matrix. And they'll continue reproducing. New bacteria will join that matrix to make this final kind of larger biofilm community. And then at some point, either individual bacteria or actually maybe clumps and groups of bacteria will leave the biofilm to go off and kind of start that whole cycle, recolonize and start that whole cycle again. And so that's the biofilm life cycle just in general. In the classroom, we work with this safe bacterium known as Pseudomonas fluorescens. So this is just kind of covering a little information about the bacteria that we work with. It was isolated on sugar beets, and it actually protects the beets from fungal predation. So it helps kind of a commensal relationship with the sugar beet. These are actually images, scanning electron microscope images that we were able to take in our lab. So that's just one single individual bacterium these bacteria have flagella to help them move around. And so that's a flagella that's still attached. And then this is a whole community of little individual bacteria that are encased in that extracellular slimy matrix. And this is just an image of bacteria that have been streaked out on a plate. So you can kind of get to where individual colonies are, but you can also see that if you put them under ultraviolet light, they glow, given the name fluorescens, right? And this is just a little bit of a more detailed overview of why we chose this bacteria. It's safe for classrooms, it's probiotic. It's already fairly good at forming biofilms, which helps it to stick to plant surfaces, and typically forms those smooth round colonies when plated. That's what the ancestor forms. And just one interesting fact is that the biofilm matrix is composed of a lot of different materials depending on the bacterial species. So usually it's like sugars and proteins and DNA. The Pseudomonas fluorescens matrix is primarily made up of cellulose, which is pretty interesting because it's a polymer of glucose, and it's the most abundant organic polymer on Earth, and it also, if you like plants, the main component of plant cell walls. So that's kind of provide a little overview of the bacteria and how it survives in the natural world. And in the laboratory, what our lab has come up with is I think a really innovative way to replicate looking at how bacteria adapt to this biofilm life cycle by using just plastic beads. And so what we can do is put a plastic bead, a sterile plastic bead into a test tube, and then inoculate that test tube in the media with bacteria. And we allow that to grow, the bacteria, overnight. And bacteria will reproduce in the test tube, and some of them will attach to the bead and form biofilm, right? And so the next day, then we add a new bead. These beads are a different color just because as you move along through the experiment, you want to know which bead came from which day you were following. And you add the biofilm covered bead to the new clean bead with clean media, wait again another 24 hours. And then what's going to happen is biofilm, the bacteria that were forming biofilm on that original white bead, some of them are going to leave the bead to attach to the new bead and form biofilm there. And so what we're doing is putting these bacteria under pretty strong pressure because we're only moving the beads. And so those were the bacteria that were best able to colonize the bead and form a biofilm. And this is an overview of how we select for the biofilm, but so classrooms will do 2 replicates where they're doing the bead transfers to select for high biofilm forming bacteria, but also as kind of a control, compare those to bacteria where there's no bead present. So you're just transferring a portion of these free swimming bacteria or planktonic bacteria from test tube to test tube over the course of the week. Okay, so as you could kind of see if you went through later portions of the slide, we can actually see these populations adapt really quickly. Like the experiment starts on a Monday and really the entire experiment is done by Friday with another day you need to wait for them to grow to make observations of what the colony morphologies look like on plates. So how is that possible? Well, first off, bacteria replicate really quickly, right? So we can, so if you think about it, on the first day, 1 cell will divide to become a billion cells, and 1 in every 1,000 cell divisions is going to have a mutation, which is actually a fairly low mutation rate. But but even given that, because you have so much replication, we're able to sample, all the mutations are happening randomly, right? But we're able to sample millions of mutations across the population every day. So you have millions of mutations, most of which, of course, are going to be bad for the bacteria, right? Those bacteria, you know, will probably die off. But some of those mutations might allow the bacteria to be better able to attach to the bead and form biofilm. And so that bead transfer isn't random. That's a force of selection. And so some of the mutants that are better suited to being biofilm formers, we're going to select for them, and then the frequency of those mutants is going to increase in the populations. Oh, and so here's where we go to set 1 of discussion questions. So I'm going to, yes, add these to the chat room. I don't know if I can see. Maybe I'll just stop the share for a second. Okay.
Are these somewhere on Google Classroom, Abby, that we can point them to?
They're also, yes, they're also in Google Classroom. So I'm trying to just drag and drop them into the chat, but that didn't work. Yeah, I can, let's see, add a file. Oh, there you go. I can also put them in the chat.
Are these the questions from the Google Doc?
Yes, in the classroom.
So I just put the link to the Google Doc. And what questions should they look at in this first discussion?
So I actually changed them a little bit. Sorry.
Ah, okay.
Sorry. Right. Too much technology for me. I can. So under Evolving STEM Introduction to the Experiment, there are introduction question sets.
Mm-hmm.
And so the first set is question set 1.
I am not seeing that. The only thing I see under the introduction questions is the Google Doc that I just pasted.
I'm not sure.
Oh, wait, introduction discussion question set. I got it.
Sorry.
Okay.
So there's one of questions, which just kind of stops at this point, but it's the same ones that were just up on that PowerPoint.
Okay, so this is set 1. All right, so let's try this document instead. So hopefully everyone can go to that or look on the Google Classroom. And I think what you're doing, Abby, we're going to break them into small groups and give them 5 to 10 minutes to talk among themselves about some of the answers and then come back to discuss.
Yeah, so if you guys, yes, so we'll break you up into small, I'm not going to do that, but we'll break you up into small groups. So just discuss the questions and come up with some answers, and then maybe nominate someone to take the lead on sharing what you thought with everybody else, if that's okay.
That sounds great. Solomon, do you want to break them up into rooms? And Solomon, you're muted.
Sorry, just making sure that everyone's paired off properly.
Okay. Does anyone have any questions while Solomon is transferring you to rooms? Was everyone able to access that link? Here, I posted also what the questions are.
Oh, and I did try, some people, thank you very much, added some discussion questions. So if I didn't feel like our questions covered them, I tried to add them in to the discussion. So, or to the questions that are posted.
Should be ready now.
And there they go. And Solomon, I think as soon as we come back to the big group, you can start setting up the breakout rooms beforehand. You don't have to wait until we say we're ready to breakout rooms. So you can set that up while Abby's talking too. So we don't have that couple-minute lull.
Right.
You just can't open the breakout rooms or they'll all go away. Because you can even set them up beforehand too. But of course, we don't know who's going to be on the call. But you should be able to do it while Abby's talking.
Yeah. I'm able to do it. I just forgot that we had to separate the some of the deaf students and some of the other students.
Solomon, can you put Daniel in the room with Sarah?
Oh, sure. Does anyone else need to go into one of the rooms?
Okay, right now. I think in a minute or so, we should try to pop in and out of rooms to see if they're discussing or have questions. How many rooms did you put them in, Solomon?
3.
Okay.
Each room with 3 to 4 students.
Great. So we'll see how this goes. I really wish that more than one person could control breakout rooms.
Yeah, that's a weirdly annoying feature. I think just the way it's coded there is that like only the host can do it, and so whoever is not first to open the room, even if it's the same account, isn't the host. Because like when I log on with like the laptop and the main my main computer, it only says one of them is like officially the host.
Right. Yeah, I mean, likewise, I log into the same account, our Hillman Academy account, but am not the host.
Right.
So Solomon, do you want to, and I think that we'll be able to leave the breakout room, but you're the only one who can put us in them. Do you want to put Abby and I into 2 different rooms for a minute, just so we can check in to see how they're doing, if they have any questions?
Sure. I'll put Abby into one and then you into the second one.
Perfect.
How many breakout rooms do we have? Just 2 or
3.
Okay.
So we'll come back and then hop back into the third one.
Mm-hmm.
Whoever comes back first. All right. Hey, Solomon. Has Abby been back yet?
Nope.
Okay. Do you want to put me into the last breakout room?
Sure. Weird. Oh, okay.
Breakout room 3?
Yeah.
All right. All right. Still nothing from Abby, Solomon? So Solomon, the other thing that I can't do because I'm not the host is send a message to everyone. Do you mind sending a message and telling them like 3 more minutes? One group was already finished. The other group was about halfway done that I was in. Thank you.
All right, should I close all the rooms, give them the last minute?
Yeah, that'd be great.
All right. Looks like everyone is out.
Everyone's back. Great.
We're having a really interesting conversation about stuff. So maybe before we move on, if people would like to share what they discussed in their breakout sessions.
So is there any specific question that any group was discussing where there was either confusion or interest you'd like to bring up? Like I know in group 2, I think it was group 2, you were having some conversation of what's the purpose of, or what does the experiment test? Do any other groups want to talk or want to answer what do you think that this experiment tests? Maybe we can ask someone from, so Abby, you said your group was having a lot of discussion. I'm not sure who was in the groups. This is another hard part of being in Zoom. Who was in group 1? Does anyone want to discuss anything?
I could
Yeah, go ahead, Amanda.
So like it tests like you're selecting for the bacteria that can adapt enough to form like a biofilm on these beads compared to a control of free swimming bacteria that don't have any beads to form communities on. And you're like, by the end of the experiment, you're testing to see like the amount of communities there were, or like maybe any mutations that you saw in like phenotypic mutations.
Yeah, I think that's a great answer. Right. Yeah, so we're putting with the beads, we're putting bacteria in a new environment where we're forcing them to attach and form biofilm communities on these beads. And both the bead and the planktonic populations, there'll be random mutations are random, right? So random mutations will occur throughout the bacteria's DNA. In the case of the bead populations, some of those mutations are going to make those bacteria better able to maybe attach to the bead more quickly, or outcompete other bacteria that are attaching to the bead. And as we select, and since we're only moving those bead covered bacteria, we're going to increase the frequency of those bacteria that were better biofilm formers over the course of a few days. And then we can see that they're better biofilm formers because they look different when we plate them as colonies. There are 2 ways actually we can see. We can watch increased biofilm formation on the sides of the test tubes over time, and we can also plate the bacteria, and then just from our previous knowledge, we know that these changes to the colony shape go along with changes to their ability to make more biofilm. So does anyone want to ask additional questions before we kind of move on to the next little section where we dive in a little bit more of what the daily experiment looks like?
I have a question, but I'm not sure if it's something that you guys will talk about later. I was just wondering if you guys have ever like tried to extract like a plasmid from the new bacteria and compare it to like DNA from the ancestral bacteria and like see what exactly like sequence that DNA and see what the mutations were? Like, is that even possible?
That is the best question ever. Yes, so not plasmids, but the actual bacterial DNA. Yes, we've extracted DNA from mutant bacteria and ancestral, like their ancestors, and we can compare them. And we actually look at the whole genome of the mutants and compare that to the genome of the ancestors to see where did that mutation occur in the mutant. In which gene, how did it change that gene? And then what does that mean? Like, why do we think that caused increased biofilm formation? And you guys, that's gonna be the fun project that's coming later, is that we have sequences and you guys are gonna be able to do that bioinformatic analysis to compare mutants to ancestors and figure out where the mutation was and then some information about what it was doing.
I have a question. I think you mentioned this a little earlier, but can you say again, like why we moved them from test tube to test tube?
Oh, sure. Well, one thing is that the bacteria they'll need more food, right? We'll need to replace their food source. So you need to kind of transfer because they'll consume all the nutrients and stuff in the original test tube, and so you need to provide them with new nutrients. So that's the transfer gives them fresh media, fresh food to eat, and then it's also putting on like another for the beads, another pressure to keep forming, to keep completing that whole life cycle of attachment and biofilm formation.
That makes sense.
All right, I think this is good that we'll probably end up splitting this over 2 days, but I will try to go through this next part a little bit more quickly. Okay, so we went through these questions. So the next steps, and I'll kind of run through this a little bit quickly because I think you guys were discussing this in the last part too, but this is just kind of what was the daily experiment. If you haven't had a chance to watch these videos, they really do give a nice, you can watch people performing the experiment on their own to kind of get a better idea of what it looks like when you're doing it. But essentially, and there's a lot of text, but this covers kind of like, what's the protocol? What do you do when you're in the lab? And so you add beads to culture tubes on the first day, and then you inoculate that liquid culture with an individual colony from a plate so the ancestor has been streaked out across these plates, and you pick an individual colony, which should represent just one genotype for the bacterium, inoculate the test tubes, and then we grow them overnight. I think someone had asked a question before, what's the conditions? We actually typically grow them, you can grow them a lot of different ways. These bacteria like to grow between like 25 and 32 degrees Celsius. 28 is their most preferred temperature. So we'll grow them in an incubator that can maintain that temperature. And we grow them on a shaker to oxygenate the media. So shaking tubes, and since they're in an incubator, it's usually in the dark, but they can be grown in light also. Oh, and there's the video. So yeah, so then the next day after they've had a chance to grow up overnight and attach to the bead, we add a new bead, or okay, so first what we'll do is take the bead from the overnight culture and we actually put it into these tiny little Eppendorf tubes, and then vortex them. And we talked about that a little bit in my group. So why vortex? A vortex just agitates the culture really strongly. And we're doing that to get the bacteria off the bead, right? They're good biofilm formers, they're good at attaching to the bead, and so we want to make sure that they all come off the bead and are released into the liquid media so that we can then first transfer them to a new tube with new media to kind of keep that population going. And then the other thing we do on this second day is we dilute the bacteria multiple times. So that's what this is, a dilution series. And then plate the more diluted cultures. And I think this is a question that comes up later, but maybe I can just ask it to you now while we're going through it. So why would we want to dilute the bacteria after, so we've vortexed the bead, we've got a bunch of bacteria in this original tube, and then we're gonna do a serial dilution. So yeah, do a few dilutions and then only plate the most diluted portions of those bacteria. So can anyone imagine, why would we want to dilute from that original culture?
If you don't dilute it, there will be too many bacteria colonies, and it will basically create a lawn on the plate. You wouldn't be able to see each individual colony morphology.
That is the exact correct answer. Yep, we want to dilute them because there'd be way too many bacteria in the original population. There are a lot of bacteria in the tubes. So we dilute just so in the end we can have enough bacteria that we can see, get a snapshot of the population, but also that they're separated on the plate and we can see how the individual colonies grow. So the next few days, if you were doing this experiment for the week, are really just then transferring the beads. And the point of diluting and looking at the bacteria early in the experiment is to see, what did the ancestral population look like? You know, what did you start with and what did they look like when we plated them? So then we do a few more days of bead transfers. And then at the end, we just take those bacteria after we've done these few days of selection and plate them again. The bacteria take a few days to the colonies to grow up so we can visualize them, see what they look like. So you actually, you can make observations on this first Friday about what the ancestral populations looked like, but you won't be able to make the final observations until Monday. And maybe just to point out here, so these are the two different dilutions. So this is the 10 to the minus 2 dilution and the 10 to the minus 3 dilution. And this is for one experimental population. So you can see the higher dilutions have fewer bacteria and you're better able to see individual colony morphologies. And then this is the control. And really the other thing to point out here is if you compared these, all of the bacteria look the same. Whether they came from the bead or from the control populations with no bead, they're all just gonna still have this smooth circular shape because we're just looking at bacteria really that we just grew for one night. So then you can wait for the weekend for those final bacterial colonies to grow up and look at, now have these populations changed, right? And so on the top here, this is from a real experiment. Another, I'll mention, if you read it carefully before, we plate two different dilutions, so one more and one less concentrated. And that's because in some cases, maybe you didn't get a lot of bacteria. You want to be able to see some. So, and you can see in this experiment, you know, I didn't get too many bacteria on this plate, so it's hard to really know what that whole population looks like. But in general, these first two, so these were different experimental populations that were selected to attach to the bead. And if you look at these carefully, you can see most of the colonies still have these little, they still have that circular smooth shape, but this guy here, in this population looks different. It's kind of this little roughly thing that has a different appearance than the other colonies. And in this other population, it's pretty highly diverse, right? Where you have these big spreading wrinkly colonies, and you can compare those also to bacteria that made little tiny colonies in comparison to the ancestor. And if we compare that to the controls, we can see that in this case, all of the controls still maintained populations that only have these smooth ancestral shaped bacteria. So this is the next set of discussion questions. I don't know if we have, if we want to break out for this, or do you want to try to answer it just as a big group? I know we already talked about vortexing and doing the dilutions. And I guess essentially, what does plating reveal? What does the population look like, the overall composition of individual bacteria? And I thought a good question that someone posted, so if anyone would like to think about this right now, is how might other species of bacteria behave in a similar experiment?
I guess since not all bacteria are the same, some other species might have different shapes, or some of them just like might not be that good at forming biofilms as the Pseudomonas fluorescens. So I think it just like depends on what type of bacteria it is.
Yeah, yes, exactly. That's what I would say. It depends on the bacteria. We know, if you remember from the very beginning of the experiment, we actually, our lab originally looked at a different species, Burkholderia, right? And they don't make the exact same shapes, but do have similar, similar changes to colony morphology when selecting for high biofilm formation. People have also looked at that in E. coli, but yeah, it's going to depend on the bacteria, how they behave, and in the end, what they'll look like and how quickly they'll adapt to making biofilms on these beads. So I think it's both species dependent, but we know that there are other bacterial species that will behave in a similar way to what we're looking at in the experiment. Yeah, and this is just to show that over time, typically if you look at these day 2 cultures, you really can't see a lot of biofilm formation on the side of the tube. But after a few days of selection, a lot of especially if you think you're getting some high biofilm mutants, you can kind of tell in advance, because when you look at these tubes, you can see this kind of layer of biofilm formed on the evolved population tubes. So that's one way to look, and another way to look at how the populations are changing are those individual colony morphologies. So this is just kind of zooming in. So here's plated on day 2, all of them, all the bacteria are making these smooth circular colonies. This was an especially diverse plate, but you can see multiple different phenotypes appear on the plates with these kind of large spreaders and the tiny ones that are little wrinkly guys, and ones that are kind of in between too, right? So, so you can see these changes in diversity over time in 2 different ways with this experiment. So maybe if we think, I know we're kind of at the end of the hour. But maybe we can try to do one more question before we break for today, if that's okay.
Sure.
I'm gonna
It's a question on Classroom. I could grab it quickly.
I
Oh, you already did it. Perfect.
So this is kind of a long question to think about and a little of a thinker. So maybe you just, if you break up into your groups, you know, think about, you know, why populations might look different, essentially, at the end of the experiment.
Okay, so we can break them into group and give them about a minute to discuss, and then we'll bring them back in case, Abby, you want to have a discussion for a minute and then set them up for tomorrow of what they'll need to do and expectations for tomorrow. That work? All right, Solomon, you want to send them off?
All right.
All right. I don't know why there's always one that seems to lag.
It looks like it's almost always the same. I wonder if it's just like a computer issue.
Maybe. All right.
So I think looking, we're probably like halfway through the first slideshow, and it seems like people are having good questions, and I feel like, especially with kind of medical relevance, people seemed interested in that as far as the few questions that came back.
Mm-hmm.
So I'm thinking, let's just tell them, you know, no, there's nothing new for tomorrow. If you know, just go back through the stuff from today. We'll go through the rest of the slideshow tomorrow, the second half, and do the same kind of breakout. Yeah, and kind of just finish up this introductory, like, what was the experiment like?
All right, I think that works. And when we break them out and bring them back together, if they have multiple questions, I don't know if you want, so that it's consistent across groups, I don't know if you want to give each group a single question to think about. That way it could take less time and they can come back and we can talk about all of the questions as a large group. That way everyone gets to hear kind of the answers to all of them in case any of the groups work. So sometimes that works. Or if we just need more time and we come back together to kind of talk about everything too, I think we'll be good. We only have about a minute left though, Solomon. You want to kick them back out so they come back?
Sure. I'll just close the rooms right away.
The other nice reason to do that, Abby, is when they come back, then they know that they'll be responsible to answer one. And then you don't have that like awkward silence too, because each group will know, I need to answer this and we've talked about it. And then they'll just do it.
You know what, and for this question I just gave them, which is a little bit of maybe a big one, if they want to keep thinking about that for tomorrow, we can.
Yeah, that could be where we start tomorrow too.
Yeah.
So you can just tell them that and set them up for what they need to do tomorrow.
All right, everyone's back.
Okay, great. So we're just talking about, I know that this question is kind of a longer one and something to think about. So I think what we'll do is for tomorrow, you know, think about this kind of discussion question. I won't post any new material for tomorrow. So, you know, you could review kind of that first half of the slideshow that we went through today, or go through the second half, and we'll kind of start into that tomorrow and continue with the same, you know, take little breaks, have discussion on different aspects, and then come back together. And then we'll just move some of the other materials that we're thinking of doing later in the week, which I think some of them they're shorter, so I think they won't take so much time to get through, if that's okay. So does that make sense? No new material. Just review what we've looked at today, and we'll plan to come back tomorrow, finish the slideshow, and keep up the discussion.
Sounds good. Does anyone have any questions for Dr. Matela before we leave? All right. Well, remember tomorrow also at noon, we will have our second journal club. So I look forward to that. So I look forward to hearing about research progress and papers that have maybe helped form your research projects. So I'll see you guys tomorrow at noon.
All right.
Thanks, everyone.